Hello, everyone. Yeah, like we know a lot of the delivery of nanoparticles, they based on needle-based injection. And here we want to do something different. We want to developing a needle-free technology to deliver drug. And here I'm presenting some work about sublingual delivery of proteins. Here is the land acknowledgement. And Dr. Lee is the co-founder and CSO of the Nanostar. And the question comes up, why proteins? We can see the size, the market size of protein will reach 461 USD million in 2029, which is almost double to 2022. The annual growth rate is around 8%. And North American leads the protein markets. Among those protein drugs, most of them are insulin, and then around 30 to 40% are monoclonal antibody. So our aim is to deliver a wide molecular weight of proteins from insulin to monoclonal antibody. Those proteins have been widely used for chronic diseases such as diabetes and cancers. However, because the molecular weight of those proteins, they are big, so they are very difficult to penetrate the cell membrane. They are unstable, so they cannot be stable in the GI tract. Most of them have to rely on needle-based injection. However, for people with chronic disease, if they do the regular needle-based injection, such as diabetes patient, they need to receive insulin like several times per day. So it means you need to be poked several times per day. It will be very painful. So what we try to do is release them from this kind of painful injection. But by using needle-based injection, patient usually has a poor adherence. For example, for diabetes patient, the medication adherence for insulin is only around 30 to 60%, which may result another kind of side effect, such as ulcer. So here we focus on sublingual delivery. Compared to oral injection, administration, sublingual environment is less harsh and is able to bypass the first-pass metabolism. And when we do sublingual administration is under the tongue, we need to drop a sublingual formulation here. However, before those drugs get into the blood circulation, there are a few barriers we need to overcome. The first barrier is the mucus barrier, and the second barrier is the multiple epithelium layers. After that, drug can reach the blood vessel and then get into the blood circulation. There are a lot of technology have been developed to enhance the penetration through this mucosal, such as mucosal adhesive materials or some penetration enhancers, such as surfactant. However, this kind of materials either raise the issue of safety or they has low permeation ability. What we focus now is using cell-penetrating peptide. Cell-penetrating peptide is safe. They consist with amino acids, which can be degraded in our body. And they can translocate the cargoes from outside the cell and get into the cells and become functional. In our previous work, we figured out once clinical approved cell-penetrating peptide protamine, it can use to enhance the penetration of of BSA, the model protein we choose. It had been labeled with FITC, a fluorescent dye. We figure out protamine can enhance intracellular delivery of BSA compared to RA peptide. RA is a widely used peptide that consists with arginine. And interesting, we find that in RA delivery group, most of the protein being delivered have been trapped into the lysosome. However, when we're using protamine, we can bypass the lysosome pathway. In our further investigation, we figure out protamine can aggregate on the cell membrane in a punctate member. This kind of aggregation can help to permeate the cell and then get those proteins inside the cell. So the idea here is, can we modify the protamine to developing some nanostructure and then further permeate the cells? Because we want to do sublingual delivery. In sublingual, there has a shorter retention time. Those drugs need to be observed quickly so that they can avoid the swallow of patient. So here we're developing two normal CPPs, CPP1 and CPP2. CPP1 is a nanoparticle and CPP2 is a nanorob. The structure has been confirmed by TEM and AFM. After that, we're using human primary sublingual keratinocyte as the model cells and then treated them with different CPPs. In the untreated cells, we can see the surface of the cell is very smooth, and in protamine, we can see some aggregates of protamine. And in CPP1 and CPP2, we see more aggregation, and we can also see some pore formation under those CPPs. To quantify those pore formation area, we do the quantification, and we confirm that normal CPP forms more pores compared to protamine. So now we know that this nanostructure can form pores on the cell membrane. We are wondering if this pore formation can enhance the penetration of proteins. Here we choose IgG as protein as our model protein with a molecular weight around 150 K. And we can see here the protein has been labeled with the red signal. In the normal CPP group, we can see the increased uptake of this red signal compared to protamine, and they are able to bypass the lysosome pathway. Because we want to deliver those proteins into the through the sublingual route. In sublingual route, it has multiple layers. So here we're developing a multiple layer model, a spheroid, to see if this normal CPP can help those IgG to penetrate, overcome those multiple layers. Here is a spheroid. If the penetration ability is good, then they should be able to permeate those cells and help the penetration of the IgG from the periphery to the core. The distance is farther. It means that the penetration effect is better. And we can see that in the normal CPP group, the distance of distribution from the periphery to the core is around 70 micrometer. And in the protamine is around 50 micrometer. So we confirm that normal CPP has a better penetration efficiency compared to protamine. After playing around with the 2D model, the 3D model, we want to know what our technology can behave in the in vivo. So we apply this technology in a mice model. Here is the sublingual tissue collected from mice. And from here, we can see, from the IgG-only group, we can barely see the red signal. In the protamine-treated group, we can see the red signal here. They are on the top of the sublingual tissues, which is the mucus layer. The protamine can barely overcome the mucus layer. However, in the normal CPP, in the CPP1, we can see the increased intensity of the IgG. We can also see some penetration of IgG in the epithelium layer. We see more penetration of IgG in the epithelium layer in the CPP2 group, which confirms that this normal CPP can enhance IgG penetration in vivo. After that, we want to know if this normal CPP can be used to deliver some therapeutic protein, so have a actual therapeutic effect. Here we choose insulin and semaglutide to control the blood glucose level of mice. We first test insulin. Here, when we look into the red line, when we deliver insulin by using protamine, it can barely decrease the blood glucose level. However, when we're using our normal CPP, they can decrease the blood glucose level since 0.5 hours, and the lowering BG effect can be maintained for 6 hours, which is almost comparable to the sub-Q injection. So from this figure, we can know that our needle-free technology can deliver insulin to achieve a comparable effect as the needle-based injection method. And next, we try the semaglutide. Here we challenge the mice with the glucose first. After mice be challenged with the glucose, their blood glucose level should be go up very quickly. However, if we deliver semaglutide successfully, then it should be able to suppress the increase of BG. From here, from the protamine group, we can see the BG have been suppressed since 40 minutes. But in the normal CPP1, it suppressed the BG since 20 minutes. And after 24 hours, we re-challenge those mice with glucose again. And there is almost no fraction in the normal CPP group, which confirms that the normal CPP has a better effect to control the blood glucose level and can deliver semaglutide more efficiently compared to protamine. Because we aim for deliver a wide range of protein drug, such as monoclonal or antibody. So here we try another therapeutic drug, such as human growth hormone. It weighs a molecular weight about 22K. From here, we can see that the normal CPP, they can significantly increase the hormone growth hormone in the plasma compared to the hormone growth hormone alone group. And the result is also comparable to its sub-Q preparation. So it means that our needle-free technology can deliver human growth hormone in a comparable way to the needle-based injection method. And next, we try to deliver the monoclonal antibody. As we can see here, the normal CPP can enhance the plasma concentration of IgG compared to IgG only group. However, when we compare it to the sub-Q, to the needle-based injection method, it's still not very encouraging. The needle-based method still is the best one compared to this needle-free technology when we try to deliver the monoclonal antibody. However, it already achieved a very encouraged amount of AUC compared to the IgG only group, which means that if we want to achieve the similar effect as the needle-based injection method, then we need to do more optimization for this cell-penetrating peptide. And we can also think about the other kind of diseases, such as we can because we already observed the penetration ability of this normal CPP to deliver monoclonal antibody to the epithelium layers. So we may focus more on those epithelium layers disease, such as chronic rhinosinusitis with nasal polyps. So yeah, here may be the future plan of this project. And I would like to acknowledge my supervisor and the lab and our cooperators. Thank you.
Thank you very much, Jiamin. Are there any questions? Oh, over here. Yes, speak really loudly. There you go.
I do have the following questions. Is there any chemical conjugation between the CPPs and the proteins, the drugs of the tribe, or the larger payloads, or they just go-administered?
Good question. There is no conjugation between the CPPs with the drug we delivered. They just a simple mixing method. Yeah, we just physically mix them together. They do not have any chemical conjugation between the CPPs and the payloads we delivered.
If I can have a follow-up question to that, are they encapsulated in the nanostructures, both of them, or just the CPPs and the drug is outside of the nanostructures?
They are outside the nanostructures. Yeah, but there maybe has some charge interaction between the CPPs and the drugs. Oh yeah, it's a good question because we are submitting these papers and we do some modification on those CPP. They are derived from the protamine.